Imagine a 4-kilogram salmon lying dead in the forest, dozens of kilometers from the sea, and from its body, a spruce tree lived another 300 years. This is not mythology, not metaphor. It has been measured in radioactive isotopes locked inside the growth rings of ancient trees standing beside salmon streams. A single salmon carries within its flesh nutrients the forest cannot draw from soil alone. The nitrogen density of a salmon run rivals the fertilizer load of a small city. How does ocean protein find its way into ancient forest? And why are forests without salmon so measurably poorer than those that have them? The answer begins not in the forest, but in the exact gravel stream bed where a salmon was born and to which, against all odds, it will return. Pacific salmon are not one species, but 5 each, with its own name, its own season, its own river, making the salmon run a year-round pulse rather than a single event. We begin with the sockeye, the most numerous, the most studied, and the species that has taught scientists the most about what happens when the ocean comes to the forest. Each sockeye spends 1 to 2 years in the freshwater lake where it was born, its body gradually rewriting its own chemistry before the ocean will accept it. This transformation, called smoltification, rewires the fish's entire salt regulation system, driven by the thyroid and adrenal glands working in a biological crescendo that lasts weeks. Out in the ocean, the salmon feeds for 2 to 4 relentless years, small fish, krill, squid, accumulating the fat and nitrogen of an entire marine food chain within its own body. By the time it is fully grown, over 95% of its body mass came from the ocean. It is, in the most literal sense, a parcel of sea delivered to the mountains. Then the strangest thing of all begins. The salmon stops eating entirely, turns toward land, and begins the most extraordinary journey in the animal kingdom. For the entire upstream journey, sometimes weeks, sometimes hundreds of kilometers, through rapids and rock, the salmon does not eat a single thing. Instead, the fish begins consuming itself, mobilizing stored fat and protein to fuel muscles and eggs in a process from which there is no return. It is running on borrowed time. This is what separates Pacific salmon from most other salmon on Earth. They die after their single act of reproduction, without exception, without return. Their death is not a biological accident. It is a mechanism refined over millions of years, specifically to deliver ocean nutrients into the forest, an evolutionary gift to the land. The moment the salmon fall, the forest begins its work, and the first to arrive are not trees, but the largest animals in the entire ecosystem. A coastal brown bear can consume up to 90 kilograms of salmon per day during the peak of the run, and even they, in their legendary appetite, cannot eat it all. What matters is not what the bear eats, it is what the bear carries. Up to 200 meters from the stream, beneath the canopy, the bear leaves behind what it cannot finish. At sites where bears regularly carry salmon inland, scientists have measured soil nitrogen levels up to 30% higher than at comparable sites without bear activity. But the bear is only one link. Simultaneously, dozens of other species are engaged in the same work, each carrying ocean protein deeper and deeper into the forest. Bald eagles, ravens, kingfishers, wolves, cougars, mink. Each carries salmon in its own way, weaving an unplanned distribution network that no engineer could have designed. And the carcasses no one claimed, the stream itself, becomes the final courier, dissolving their nutrients into the water table beneath the riverbank trees. Along a single kilometer of stream in the British Columbia interior, an estimated 75,000 kilograms of salmon die each autumn, the rough equivalent of 1,000 cattle laid down in the forest. But this is only the surface of the story, because the vast majority of salmon protein never travels by bear or bird, but follows a slower, invisible route into the heart of the forest. Those threads you see under the damp forest floor are not the mushroom, they are its roots, and they extend for kilometers beneath every step you take in an old-growth forest. Every forest fungus maintains a mycorrhizal network, its threads fusing directly with tree roots, exchanging nutrients, sugars, and even stress signals across the entire forest floor. Canadian scientist Suzanne Simard spent 30 years studying this network and proved that the oldest mother trees use it to nourish seedlings growing in their shade. But the mycorrhizal network does not only carry sugar from tree to fungus, it carries everything, including the nitrogen locked inside a dead salmon, through a long and slow chemical chain. The first step is decomposition, and as the salmon lies on the forest floor, hundreds of bacterial and fungal species begin dismantling its complex protein architecture, piece by invisible piece. Black flies arrive within hours of death to lay eggs, and their larvae can reduce a large salmon to bones in under 24 hours. A biological demolition team that works without rest. But the larvae are not only decomposers, they become food for dozens of bird and insect species. And when they die, the nitrogen spreads another step outward through the ecosystem. Each step in this decomposition chain is a transport step, moving nitrogen from its locked form inside salmon protein to its soluble form in soil, where tree roots can absorb it directly. This is where ocean nitrogen begins its upward journey, not on legs or wings, but through the invisible water columns inside the living tree, drawn upward by the sun. To understand this fully, we need to look at one scientific technique that completely changed how ecologists see streamside forests, a method that reads the past inside the present. Every nitrogen atom on Earth exists in one of several isotopic forms, and ocean nitrogen carries a distinct isotopic fingerprint, different from terrestrial nitrogen, measurable in a laboratory. When scientists cored spruce and cedar trees along salmon streams in British Columbia, they found the isotopic signature of the ocean, embedded inside the wood itself, ocean nitrogen written in tree rings. Most striking of all, the proportion of ocean nitrogen in the wood spikes in rings that correspond to boom years for salmon runs, and falls in years when the runs were poor. This means each tree ring records not only the weather of its year, but also the abundance of salmon that died in the stream beside it, a dual chronicle of ocean and atmosphere. A streamside spruce in British Columbia can grow to 80 meters and live for 1,000 years. And within those 1,000 rings, this is the point ecologists John Helfield and Robert Neyman established in 2001, a finding that reshaped everything about how we understand the ecology of streamside forests. They measured tree growth along salmon-bearing streams and compared it against tree growth on streams without salmon, holding all other variables, constant temperature, rainfall, soil type. The result was unambiguous. Trees beside salmon streams grew up to 3 times faster than trees beside streams without salmon, most dramatically within 50 meters of the water's edge. 3 times faster, to give that a human scale, means a sapling that would reach your roofline is instead reaching the height of a 3-story building in the same span of years. And faster-growing trees don't simply mean more timber, they mean a darker, cooler, moister forest, creating better habitat for the very streams whose salmon are feeding them. If salmon feed trees, and trees shade streams, and cool streams raise healthy salmon, then this is a positive feedback loop, a self-amplifying system of staggering elegance. To understand the mechanism in more detail, we must look closer, not at bears or birds, but at the smallest inhabitants of the forest floor, the ones invisible to the naked eye. In 1 teaspoon of old-growth forest soil beside a salmon stream, there are more bacteria than the total number of human beings who have ever lived on this planet. Many of these are nitrogen mineralizing bacteria, converting organic nitrogen from decaying salmon into ammonium ions, then into nitrate, the form that tree roots can directly absorb. This process is called nitrogen mineralization, and it peaks in late autumn, precisely when the forest floor is densest with salmon carcasses, a biological synchrony refined over millions of years. This timing is not coincidental. Trees demand nitrogen most intensely in late summer and early autumn to prepare for winter dormancy, the precise moment the salmon arrive with their cargo. Trees don't always wait for the long bacterial chain. Their mycorrhizal root partners can directly absorb organic nitrogen as amino acids, bypassing several steps and shortening the journey from fish to leaf. More remarkable still, different fungal species specialize in different nitrogen forms, some targeting organic protein, others nitrate, creating a harvesting network of extraordinary efficiency from a single salmon carcass. Consider this: a 4-kilogram salmon contains roughly 70 grams of pure nitrogen, and productive salmon streams deliver the nitrogen equivalent of several tons per kilometer of stream per year to the surrounding forest. Compare this to the nitrogen that typical forest soils receive from rainfall and bacterial fixation, and you begin to see why salmon stream forests occupy an entirely different nutritional category. But the story goes beyond counting nitrogen grams, because salmon nitrogen doesn't only accelerate tree growth, it changes the biological composition of the needles and leaves themselves. Spruce needles that have received salmon nitrogen contain measurably more chlorophyll than those that haven't, meaning they photosynthesize more efficiently at the same light level, compounding the growth advantage. A larger tree has more needles photosynthesize more photosynthesis produces more sugar for mycorrhizal fungi, and stronger fungi pull more nitrogen from the next salmon. A compounding spiral. This is one of the clearest examples in nature of what ecologists call a cross-ecosystem positive feedback loop, connecting the deep ocean to the canopy of an ancient forest in a single living chain. And here we arrive at one of the most unexpected discoveries in this entire story, something that even the ecologists themselves did not anticipate when they began their measurements. Not all salmon nitrogen enters trees through roots. A significant portion enters through the air, through the leaves themselves, from the droppings of fish-eating birds and bears throughout the forest. Fish-eating birds like herons, cormorants, and eagles deposit their droppings directly onto canopy leaves and branches, and those leaves absorb the nitrogen through their surface, no roots required. Scientists estimate that in forests where fish-eating birds nest in large numbers, up to 25% of the marine nitrogen entering trees does so through this atmospheric pathway rather than through the soil. This means the forest canopy is not passively waiting for soil nitrogen. It is actively catching ocean nitrogen falling from above, delivered by thousands of birds in flight. And there is one more pathway, perhaps the longest and slowest, where salmon nitrogen takes a detour through the shrubs and bushes that line the stream banks before reaching the tall trees. Red alder, a common streamside shrub of the Pacific coastal zone, is an exceptional nitrogen accumulator, and its fallen leaves enrich the soil that the tall trees will later draw upon. Alder leaves fall into the stream and are carried downstream, spreading the marine nitrogen signature far beyond the stream banks into corners of the forest where a salmon has never swum. In the early 2000s, a research team at the University of Washington traced marine nitrogen signatures deep into the forest and found the ocean's fingerprint up to 500 meters from the nearest stream. 500 meters from the stream, in the deep shadow of the old-growth interior, the tallest trees still carry trace signatures of the ocean, delivered by fish that died half a kilometer away. This is the moment to pause and absorb what we have just learned. A fish dying in a stream reaches 500 meters into the surrounding forest through chains of invisible biological intermediaries. What we have understood about nitrogen is only half the story, because salmon bring into the forest not only nitrogen, but phosphorus, zinc, copper, and a spectrum of trace elements from the deep ocean floor. Phosphorus is especially critical. Mountain forest soils are chronically poor in it, leached away by millennia of rainfall, and salmon are among the most significant natural replenishment sources available. An experiment by Thomas Quinn and colleagues at the University of Washington released salmon carcasses into experimental stream reaches and then tracked the forest's response across 10 years of measurements. After 10 years, trees beside the experimental salmon reaches had grown significantly faster than controls, and aquatic insect populations in those streams had doubled. Salmon, nourishing the forest that nourishes the stream. Aquatic insects are the primary food of juvenile salmon in their first 1 to 2 years of stream life, which means the carcasses of parent salmon are literally nourishing the next generation of salmon in a direct material loop. How complete is this loop? Parent salmon die to feed trees. Trees shade streams. Cool streams raise insects. Insects raise the young salmon, and those salmon grow up, depart, and return with their own bodies as the final gift. This is not a metaphor for immortality or a philosophical meditation on death. It is biochemistry, measured by mass spectrometers and published in peer-reviewed scientific journals. But if all of this is real and measurable, why did it take until the late 20th century for anyone to think of measuring isotopic nitrogen inside streamside tree rings? The answer lies in history. For most of the 20th century, streamside forests and their salmon were managed by two entirely separate agencies, forestry and fisheries, that rarely communicated with each other. Forestry saw streamside trees as harvestable timber. Fisheries saw salmon as harvestable protein. Both missed the essential point that the two systems are not separate at all, but one. When timber companies cleared the streamside trees in the 20th century, the first thing they noticed was accelerating erosion and warming water temperatures and dying salmon runs. But what they did not recognize until decades later was that when the salmon died, the forest died with them, more slowly, more silently, in ways no one could measure without knowing what to look for. The decline was not sudden. It took generations of trees to become visible because the old-growth survivors still stood, drawing on centuries of accumulated nitrogen reserves even as the source was cut off. Once those ancient trees died and were not replaced by salmon-fed successors, the gap filled with shorter, less productive forests that looked like forests but held a fraction of the ecological capital. This raises the most important ecological question of the 21st century in the Pacific region. If forest and salmon are one system, can we restore one without restoring the other? Research from stream buffer restoration programs in Oregon and Washington suggests the answer is no. Replanting trees without salmon produces forests far thinner and slower than models predict. Even after decades, tree needs salmon, salmon needs tree, and we cannot break that circle on one side and expect to repair it from the other. This lesson cost us decades to learn, and the full bill is not yet paid. We have traveled from salmon carcasses on the forest floor to isotopic signatures in tree rings to the tragic history of 20th century forestry. But the story has one more layer, stranger and more surprising than all the rest. Scientists have recently discovered that forest trees do not only passively receive salmon nitrogen, they can, through the mycorrhizal network, actively share it with neighboring trees that are deficient. This means a spruce, standing directly beside the stream, receiving abundant salmon nitrogen, can share its surplus with a fir, standing deep in the forest interior, through underground fungal highways. Suzanne Simard and her student, Brendan Twegg, tracked radioactively, labeled salmon nitrogen, moving from tree to tree in real time, watching ocean protein travel through living wood, root by root. In their experiment, labeled nitrogen from salmon carcasses moved from the largest tree to smaller shaded trees within 48 hours, traveling tens of meters through the fungal network, crossing root systems, rising into leaves. This fundamentally changes the mental image we hold of a forest. A forest is not a collection of competing trees. It is a networked organism, distributing the bounty of the sea through its invisible underground body. The most recent research, published between 2010 and 2020, shows that this nitrogen-sharing network operates not only between trees of the same species, but between entirely different species. Western red cedar and Sitka spruce exchange salmon nitrogen through shared fungal networks, despite being conventionally described as competitors for light and soil resources in the same forest stand. This suggests that competition may be an incomplete description of forest dynamics, that at the level of biochemistry, forests operate more by a logic of sharing than of competition. This is a conceptual shift of the first order, as significant as realizing that the heart is not merely a blood pump, but the center of a whole-body signaling system. And here, we arrive at the final surprise, perhaps the strangest single fact in the entire story of salmon and old-growth forest. In years when salmon runs are reduced by climate shifts, many streamside tree species don't only grow more slowly, they alter their root architecture, growing additional surface roots to seek nitrogen from alternative sources. This suggests that trees are at some level registering the reduced salmon supply and adjusting their physiology to compensate without a brain, without a nervous system, through biochemical sensing alone. This response, documented by plant ecologist Monica Turner at the University of Wisconsin, is evidence of a kind of biochemical cognition, without brain, without nerves, without conscious memory, but unmistakably responsive to the world. And this forces a question we cannot avoid. If trees register the absence of salmon, what happens to the forest when salmon disappear, not for a single poor year, but permanently? We already have the answer in dozens of California and Oregon streams from which salmon vanished during the 20th century. The streamside forests have grown measurably thinner, sparser, and shorter with each passing decade. And thinner means less shade. Less shade means warmer streams. Warmer streams mean less dissolved oxygen. And that is how an ecosystem collapses gradually, incrementally, and ultimately past the point of return. Yet there is evidence of the opposite as well, in streams where salmon have been reintroduced after decades of absence. The streamside forest recovers faster than ecological models predicted, as if waiting for the signal. At several streams in British Columbia, where salmon returned after dam removal, streamside tree growth rates increased measurably within just 5 to 7 years, 20 years ahead of what the models had forecast. The forest had been waiting not consciously, but biochemically maintaining the capacity to respond rapidly to the return of the nitrogen source upon which it had evolved to depend across millennia. Perhaps this single fact captures the whole story best. The forest and the salmon are not two related things, but one thing, living across two different worlds, ocean and land, joined by an ancient and unbreakable bond. Return to the image with which we began: a 4-kilogram salmon lying dead in the forest, dozens of kilometers from the sea. Now we know what is happening inside it. Not an ending, but the beginning of a journey that will unfold over centuries. Inside the wood of trees not yet born, the nitrogen in that body is beginning its journey now, from protein to bacteria, from bacteria to fungal thread, from fungal thread to needle tip, and it will remain there for centuries to come. That spruce, standing at the stream edge, 400 rings deep in its heartwood, is not simply a product of rain and sunlight, but of millions of salmon that once swam this reach of river. And each of those salmon carried within its flesh a fragment of the deep ocean, of small fish, of phytoplankton, of sunlight that touched the ocean's surface years before. The Earth found a way to move solar energy from the ocean into the forest through a creature that can swim, can leap waterfalls, and knows precisely when and where to die. And when you walk through an old-growth forest beside a salmon stream, Knowing that every tree carries the ocean's fingerprint in its rings, the question that remains is not a scientific one. It is a question of what we choose to keep.